Ladies and gentlemen, welcome back to the video on the channel. Today we'll be continuing where we left off with the histology part, where in the previous video I explained the epithelial cells and the basement membrane. Today I will be explaining the junctions, all of them, and how they connect and where they connect. It's a very nice and simple topic. You can kind of consider it to be more of a snack than a full topic. You'll see why I say that in a bit. Now let's start off with the topic. Let's go into it. Starting off from this image particularly, it is a image of a full epithelial cell with the apical end being on top and the basal end being down here. We spoke about this in the previous video. Now, near the apical end, usually there is a lumen, like we talked about, and going downwards from the microvilli, ignore the microvilli for now, going downwards, mostly all of the junctions have the same or very similar order in most cells, starting off with the tight junction, then adherens junction, desmosome, gap junction, and then the hemidesmosome right before the basal lamina. This is just a general picture or general definition of the junctions and where they are, just so you can have an idea. If you guys want to go back to this photo later on during the video, just skip back and you can see it. It really helps you understand a lot of things. Now, starting off with the first one, which is a tight junction. Tight junction is also known as zonula occludens. You can kind of consider this to be a government name. Zonula occludens. Zonula means that it is bound around the cell. It goes around the full circumference of the cell. It is not just present at one point. It goes around fully the cell. So imagine this is a cell, like this whole thing is a cell. It goes around the full perimeter on the inside, obviously. And what its function is, or what it does, is that it forms a seal between one cell and between the other cell. So in this image we have cell A and cell B. Right between them we have the tight junctions, and there is a seal formed between the junctions and between both of the cells. This seal is referred to as the extracellular space. Now, why is this important? Why is this seal, or why is this region that is cut off from the outside important? Well, it is important so that we make sure that any proteins, any ions, anything that is moving from the apical to the basal, or from the basal to the apical, it moves only through the cell, not in between the cells, so that they can be processed, so that they can be digested, so that it can be taken up by the cell regardless. Because if it were to move between the cells, then a lot of those things would just get stuck in here, and it will not, first of all, it will cause a lot of gaps, and second of all, it will not be used up efficiently or necessarily. So the tight junctions prevent anything from being inside that is not supposed to be inside. Anything that the cell produces into the extracellular space is something completely different. Now, obviously, everything is made up of something else, especially in histology. That's pretty much the motto of the subject. The tight junctions are made up of claudin and occludin. Claudin and occludin. These are transmembrane proteins, which are basically these blue dots. These are the claudin and occludin. These are transmembrane proteins. They are proteins that are along the membrane, transmembrane. And what they do is that they maintain only two regions, apical and basal. They do not allow for there to be a third region, which is the lateral side, for example. Keeps it strict only to two regions, so that any molecules that are passing throughout can go into one of either region. Nothing more, nothing less. The tight junctions are very easy. That's it. One more thing to mention about the tight junctions is if you look at it under a electron microscope. Again, this is electron microscope. It gets you the details of the details. It leaves nothing up to your imagination. You can see right here this line, because like we said, zonula means that it goes around the full circumference of the cell. It is going exactly directly fully in one straight line, and you can see its structure is very sort of artistic in a way. It is like drawn together, but these are actually the claudin and occludin, the transmembranal proteins, just binding from one cell to the other. That is all that there is. Now, moving on to the next one on the list, here we have the adherens junction, also known as zonula adherens. And yes, you guessed it, zonula again means that it is bound across the full circumference of the cell, so it forms a ring around the cell. Now, the zonula adherens is right below the tight junction, and it anchors the cell to its neighbors. That is usually important for stability. So the adherens junction helps, tight junction prevents movement and keeps apical and basal only. The adherens make sure that both the cells are bound together and so that they do not move apart, so that they remain stable together. It is a bond, it's a sister bond formed between the cells. Now, as you can see in this image, we have the two adjacent plasma membranes, and again, everything is made up of something else. Here, it is made up of cadherins, which is a transmembrane, again, glycoprotein. If you've realized by now the pattern of all of them being transmembranes is because they have to cross from one membrane to the other, so there has to be transmembrane proteins. Either a glycoprotein or a normal protein, it's a transmembrane regardless. And then the cadherins, which is the transmembrane glycoprotein right here, is bound to something on the cytoplasmic side of the membrane, which is known as catenins. Now, the catenins form a plaque, as you can see here, and this plaque is bound to actin, which forms something known as the terminal web. Now, so let's break it down from the top again. We have cadherin binding to catenin, forming a plaque, binding to actin filaments, forming the terminal web. The terminal web as a structure as a whole helps in the structure of the cell, and it is a very important cytoskeletal structure. So it helps the cell hold its shape. So adherence junctions basically is important for the cells to remain together, and it's important for the cell to remain or keep its shape. That is it. That's all that there is for adherence. Again, very simple, very easy. Very demure. Moving on to the desmosome. Now, the desmosome is a very weird looking junction. It looks like an alien. It looks like something that you would see in Alien vs. Predator. Something extraterrestrial, if you may. It is also known as, it has a different name, obviously. The government name is macular adherens, and the term macula means spot, which some of you higher semesters would know because of the microbiology part. Macular adherens, which contains, again, it is made up of desmogleins and desmocollins. Now, these names are a bit more difficult than the previous one, but try to stay with me. Desmogleins and desmocollins, right here, desmoglein and desmocollins, are actually part of the cadherin family. Now, if your short-term memory is not absolutely terrible, cadherins we just mentioned in the adherin or the zonular adherens junction. So, the cadherins is a family, and a part of this family is the desmoglein and desmocollin. The desmoglein and the desmocollin both are used to form the desmosome. They are the most intracellular part, or the transmembranal proteins of the desmosome. Now, moving outwards from the desmoglein and desmocollin, they bind to the cytoplasmic end of them binds to the plaque, also known as cytoplasmic plaque, which has the government name of placoglobin. Placoglobin binds to desmoplackins, which desmoplackins then binds to intermediate filaments. Does that make sense? Did you guys get where I'm going here? Again, one more time just for a summary. Desmoglein and desmocollin are cadherins, which are transmembranal proteins which bind to the cytoplasmic plaque, which is made of placoglobin. Placoglobin then binds desmoplackin, and desmoplackin eventually finally binds intermediate filaments. It's a whole net, it's a whole structure that you will go through. Now, they are usually found as spots throughout the cell. It is not just one desmosome, there are multiple desmosomes, but they are scattered around the cell circumference, let's say. And their function, the desmosome's function, is just to provide stability for the cell. So you gotta think that the cell has its own structure, or its own structural support system, and the desmosomes carry that out, as well as the adherents. Now, moving on to the next one, The gap junctions. Now, the gap junctions, you can consider them to be the cell's walkie-talkie, or their telephones, or their cans tied to the string, whatever you want to call it. Why? It is because the gap junctions help the cells or the neighboring cells communicate with one another by sending ions, sending transporters, sending whatever it needs to send, it sends through the gap junctions. Now, everything is made of one thing or the other, and this one is made up of connexins. Now, this image right here, you can see a connexon. This whole thing is a connexon. But if you take just one singular part of this, just one of this, that is a connexin. Multiple connexins or connexins in a hexameric complex form a connexon. And the connexon is just a tunnel between one cell and the other, allowing for transport or movement of ions, of proteins, whatever is necessary. And why is this important? Why is this necessary? Why can't they just go outside normally? Well, first, because it's faster, doofus, because it goes right through the cell wall, cell membrane, because it goes right through the cell membrane. And secondly, this structure or this arrangement helps the cells in a certain tissue to work together in unison or work together in coordination. Like in the heart, when one cell beats or when one cell contracts, the full of the heart contracts. Why does that happen? It happens due to the gap junctions. It can transfer calcium from one cell to the other, allowing immediate and continuous movement or contraction, if in this example, for all cells in the certain tissue, allowing them to work together in coordination, increasing overall strength and efficiency. Felt like I was giving an ad for a car. Anyway, moving on to the hemidesmosomes. Hemidesmosomes. Now, if you remember in the previous video, we mentioned them very shortly. It was in the last image you said there's a hemidesmosome here. What is a hemidesmosome? Now, a hemidesmosome looks like, from the name, you guessed it, looks like a half desmosome. Yeah, very creative. The hemidesmosome has, instead of cadherins, it substitutes them with integrins, which binds to intermediate filaments, which binds to the laminin molecules. Right? It's very okay. Let me use this image to break it down for you, starting from the basement membrane. Now, here we have the basement membrane. Then we have the transmembrane glycoprotein, which in this case is integrin instead of cadherin. Then going upwards, we have a plaque. The plaque is necessary for the integrins, which have very low surface area, to bind to the intermediate filaments, which have a relatively high surface area. And in this case, the intermediate filaments are keratin. So, hemidesmosome is a mix of desmosome, using the structure of the desmosome, the shape of the desmosome, how it looks, but a few uniqueness, which is the laminin and the cadherin removed for integrin. That is it. And one more, let's say, honorable mention to mention. I have not placed it here because it's not really considered to be a junction. It is the focal adhesion or the focal contact, which is very similar to hemidesmosome, but instead of the intermediate filament, it is another adjustment, and we have actin. And the activation of the focal adhesion by the binding of laminin to integrin then causes activation of focal adhesion kinase, which causes certain gene expressions or mobility, and it is very common in the fibroblasts. This is just something that I thought I would mention for the future in case you need to know it. It's called focal adhesion kinase, and it's affected by focal adhesion, and it is triggered by the binding of laminin to integrin or integrin to laminin. So yeah, that is pretty much it for today's topic. Like I said, it's a snack. It's very easy, very simple. I hope you guys enjoyed. I hope you guys learned and got something new. And yeah, that is it. See you guys later. Be safe. Love you guys. Till next time.